All around us, on every single surface, and even living inside of us, there's this bustling, totally invisible world of microscopic life. But what happens when one of these tiny organisms causes an infection? How do scientists actually figure out which one it is? Well, in this explainer, we're going to become microbe detectives, and we're going to uncover the techniques used to identify these tiny culprits.
Okay, so picture this. You're a microbe detective. You've just arrived at the scene of the crime, an infection, and you've got a sample. Now, inside that sample is your suspect. But here's the catch. It's completely invisible to the naked eye. So where do you even start? How do you build a profile and pin down the perpetrator?
Well, the very first step is to get our suspects into a lineup. In the world of microbiology, this means taking that sample and giving the bacteria inside a chance to grow into something we can actually see and work with. It's a really systematic process, and it all starts with culturing.
Now, before we start running any tests, we need to know what we're comparing our findings against, right? And that's where Berkey's manual comes in. This thing is the ultimate encyclopedia for bacteria. It organizes microbes based on their genetic family tree. For our investigation, just think of it as the master file of all known suspects, our complete database of wanted posters.
So, to get our suspect ready for identification, we follow this standard procedure that scientists cleverly call the 5 I's. First, we inoculate, which is just a fancy way of saying we place our sample in a nutrient-rich medium. Then, we incubate it at the perfect, cozy temperature for it to grow. Next up is isolation. This is where we separate out individual bacteria so they can multiply into visible colonies. After that, we inspect those colonies. And finally, we move to identification, where we run specialized tests to put a name to our suspect.
All right, once we have those visible colonies, our investigation can really kick off. And the most basic starting point, the first question in any description, is super simple. What does our suspect actually look like under the microscope?
The most basic profile always starts with shape. So, our first category is the coccus, or cocci for plural. These are just the spherical or round bacteria. Super simple, like tiny little balls.
Next up, we've got the bacillus, or bacilli. These suspects are rod-shaped, kind of looking like a tiny pill capsule.
And our third main category covers the spirals. Now, this is a pretty diverse group. It includes everything from a gentle curve, which we call a vibrio, to a rigid corkscrew, known as a spirillum, or even a flexible, springy-looking thing called a spirochete.
But here's the thing. A single shape doesn't give us the whole picture. We also have to look at their arrangement, basically how they like to group together. For example, if they form long chains, we add the prefix strepto. If they bunch up in these grape-like clusters, that's staphyloco. They might also just hang out in pairs, which is diplo, or in little groups of 4, a tetrad. These prefixes add a whole other crucial layer of detail to our suspect's profile.
Okay, so we've got a visual profile. Now it's time for the single most important test in our lab, the Gram stain. This is the chemical test that divides almost the entire bacterial world into 2 distinct groups. I'm not kidding, this is a total game changer for our investigation.
So why does this test even work? Well, it all comes down to a fundamental difference in their armor, their cell wall. Gram-positive bacteria have a really thick layer of a substance called peptidoglycan. Gram-negative bacteria, on the other hand, have a much thinner peptidoglycan layer, but it's sandwiched between 2 separate cell membranes. This structural difference, that's the key to everything.
The procedure itself has 4 steps. First, we stain everything with crystal violet, which turns all the cells a deep purple. Then we add iodine, which acts as a mordant to sort of lock the dye in. And now, here's the moment of truth, the alcohol wash. In gram-positive cells, the alcohol dehydrates their thick wall, trapping that purple dye inside. But in gram-negative cells, the alcohol dissolves their outer membrane, and that thin wall just can't hold on to the dye, so it washes right out. Finally, we add a red counterstain called safranin, which colors the now clear gram-negative cells.
And just like that, the results are crystal clear. If the bacteria are purple, they are gram-positive. If they're pink or red, they're gram-negative. We've just cut our entire list of suspects in half. And this isn't just about color. This difference is clinically vital because it helps doctors choose which antibiotics are actually going to be effective against the infection.
So let's see how all these clues start to come together. We can now build a much more detailed profile, something like gram-positive coccus or gram-negative bacillus. And armed with this information, shape, arrangement, and the gram stain result, we can finally consult our gallery of known pathogens.
So let's say our tests reveal a gram-positive coccus. Our suspect list suddenly narrows dramatically. It could be the infamous Staphylococcus aureus, well known for causing wound infections, or maybe it's Streptococcus pyogenes, the culprit behind strep throat.
And what if we found a gram-negative bacillus?
Well, that could be anything from the very common Escherichia coli, which is a major cause of UTIs, all the way to the historically infamous Yersinia pestis. Yep, the bacterium that causes the plague.
The spiral-shaped suspects have their own rap sheets too. Helicobacter pylori is the agent behind most peptic ulcers, while the spirochetes include some major players like Treponema pallidum, which causes syphilis, and Borrelia burgdorferi, the tricky culprit behind Lyme disease. But, like any good detective story, there are always suspects that don't fit the typical profile. Some bacteria have these unique defenses or characteristics that mean our standard tests, like the Gram stain, just plain don't work on them.
One of these special defenses is the endospore. Seriously, think of it as a bacterium building a personal panic room around itself. It's a dormant, ultra-tough survival suit that can withstand incredible heat, radiation, and disinfectants. Bacteria like Bacillus and Clostridium use these to just wait out tough times until conditions get better.
And then we have the truly atypical files. You've got Mycobacterium, which causes tuberculosis. It has this waxy wall that completely resists the Gram stain. Then there's Mycoplasma, which breaks the rules by having no cell wall at all. And you even have intracellular parasites like Rickettsia. These are bacteria that can only survive by hiding out inside our own cells.
And just when you think you've got a handle on bacteria, it's worth remembering they aren't even the only microbes in town. There's a whole other domain of prokaryotic life called archaea. Now, they might look similar under a microscope, but they are completely different genetically, and they're famous for being extremophiles, thriving in places like boiling hot springs or deep-sea vents. It's a humbling reminder that our investigation has really only scratched the surface of this invisible world.
So, from one simple sample, we followed the clues, shape, arrangement, and that critical Gram stain to build a profile and identify our microscopic suspect. We've cracked the case for many of the usual suspects. But this brings up one last question. For every single microbe we can identify, how many more are out there, still unknown, just waiting in the invisible world for their story to be told?